Hello scientists! Today we're going to talk about the cell cycle's checkpoints. Our big idea for this lesson is that at some points in the cell cycle, the cell pauses to check if it is ready to move on. It's important that if a cell is dividing, we know that it is healthy enough or big enough, right, or ready to divide so that it'll be a successful division and we'll end up with two healthy cells that are ready to do their jobs. So today we'll highlight a few places where the cells have evolved to pause and make sure they're good before they move on towards division. Our first checkpoint is the G1 checkpoint. This happens towards the end of the G1 phase. And here the cell is asking itself if it's ready to divide. If it's going to go through S phase, right, DNA synthesis, we want to make sure that it's worth it, right? That we're going to go through and do a full division. So some things that the cell is looking for is whether there are enough nutrients so that it can go through DNA replication and the energy needed to divide, whether it's big enough to be able to divide, if it's receiving outside signals from the environment or other cells that it's necessary for it to divide, and whether the DNA is intact. We wouldn't want to go through copying DNA that was damaged in any way, because then we would end up with daughter cells that also had damaged DNA, or we might get gummed up during the S phase. If the cell meets this checkpoint, there are nutrients, it's big enough, it's getting signals, the DNA looks good, then it'll move on to S phase. If not, it will probably just hang out in G1 phase for a little longer, keep growing, keep doing its job, and it can come to this checkpoint again. Next, we have the G2 point, which happens during G phase after DNA synthesis. Here, we're about ready to divide, but we just want to do a double check to make sure that everything is good. So the big question is, what repairs need to be done? If DNA damage occurred during replication, the cell will take some time to fix it, or if it's damaged beyond fixing, then the cell might go through apoptosis, right, where the cell dies. But often the cell can fix that damage. We have all kinds of mechanisms that we've evolved to have to fix our DNA. And then another important step is that we want to know whether all the DNA replicated. If DNA replication hasn't finished yet, we're going to want to pause and make sure we allow time for that to happen. If DNA replication has gone well, our DNA is intact, then we'll move on to mitosis. Partway through mitosis, we reach the M/spindle checkpoint. I think about the M as standing both for mitosis and for metaphase, because metaphase is the stage where this checkpoint happens. What the cell is looking for is that all the chromosomes are lined up at the center of the cell, so that when they are split by the spindle fibers, half of the DNA will end up in one of the new cells and half will end up in the other. If there are chromosomes that aren't in the right spot, it's possible that one of the daughter cells will end up with too much DNA and one might end up with not enough. So we're looking to see if the spindle fibers are attached to the chromosomes at the centromere, and if we have any wandering chromosomes that haven't ended up right in the center. And if we have a problem, the cell will pause and wait for the chromosomes to get into the right place and the spindle fibers to attach. And then the cell will carry on through the rest of mitosis and cytokinesis, and our cell division will be complete. That's it for today. If you have any questions, please leave them in the comments below or stop by office hours.